even mainstream neuroscience, although they won't admit it, need entanglement. This zero phase lag gamma synchrony thing, if you look, well, nobody understands it, so we ignore it. And that's the problem. They ignore these important things that should be looked at. So I think quantum is necessary for neuroscience for a lot of reasons, including consciousness and binding. Anesthesia is selective. So it's only affecting the stuff in the quantum areas because that's where the anesthetics bind. They bind by the same quantum forces, and they're attracted, they get out of the blood and water because they're too polar, and they go right to fat and membranes, and inside proteins we have these aromatic rings. Anesthesia blocks the terahertz oscillations in the proteins in the aromatic rings.
But if it's an orchestra, then who's the conductor?
That's a good question.
Let me tell you, I've been following your work with incredible interest, and I think it's so transformative. And so maybe one way of starting this off and for our audience is give a little background on your context and give them a sense of where you're coming from. You have this notion of the yin and the yang concept of classical, and I think it's a wonderful way of framing it, classical and quantum, and consciousness is on the edge. And I think
I
that's
stole that from the Kabbalah. Somebody sent me a line from the Kabbalah that said there are two worlds, a world of strife and aggravation and a world of wonder, and consciousness dances on the edge. And I said, well, okay, the quantum classical. So that's where I got that. Yeah, thank you. I'm glad you appreciate it. I'm glad someone appreciates it.
I mean, I really want to move on that. I mean, there's another person I've had on the is Chris Fields, who's
Yeah, I know, Chris.
Yeah, I know Chris. So that'd be interesting to see how these ideas complement or don't. But I mean, Chris's notion is physics is communication across boundaries and barriers, and that the sort of the quantum classical interaction is really and a form of communication. I think is a powerful notion. Same thing. We had Don Hoffman on here. I'm sure you may know Don. So there is a new way of thinking about things that goes very deep into reframing a whole new scientific paradigm that has impact on a cultural and our cultural paradigm. And so I this is sort of the prism by which we look at it. And if you could just give a little sort of the background and what you think is the most significant things of all your findings, and then sort of where does it. You've been doing this for like 20, 25 years, right? I mean, you really.
I hate to tell you how long I've been doing. I got obsessed with microtubules in medical school in 1972. So
Whoa.
over
Okay.
years.
Well, that's that. Okay.
And then, well, so
that's a
let me
long
answer your question. So I think there's several different approaches to the current paradigm that the brain is a complex computer of simple neurons.
Right.
The first is that I don't think neurons are the fundamental units. They're not the analogy to bits. If you look at a neuron, it's very complex, and I hesitate to use complex because sometimes complexity is used as an explanation rather than a tool. But if you think of a single cell organism like a paramecium, it's only got it's one cell. It has no synapses. It's not in a network. It's a lone ranger, and it swims around, finds food, finds a mate, has sex, can learn. If you suck it into a capillary tube, it escapes faster. It finds a mate and fuses with the mate for a period of time. It is absolutely still. The only time it's absolutely still. And it's so if that's equivalent to a neuron in the brain as a one or a zero, that's an insult to neurons. If a paramecium can do that, or an amoeba, slime mold amoeba can solve the traveling salesman problem, which Adam Adamatsky has shown, among others, then it's a tremendous insult to a neuron to say you're just a one or a zero, or it's just a one or a zero. On top of that, the neuron firing is the one or the zero. And I think if we look at the neuron, that consciousness is happening in the dendrites and the soma, and the firing just reports the result. So it correlates, but it's not where consciousness is. So neurons are not fundamental, I don't think. And if you look inside the neurons for what does all that cognitive stuff, like in a paramecium, it's the microtubules. Paramecium has all these cilia sticking out from which are made of microtubules that sense the environment, that act in a coordinated fashion to swim, like oars or propellers, or in the case of a flagella. And so they're sensors and they're motor effectors. They're made of microtubules. They connect to the cytoskeleton inside the paramecium, and it's the cell's nervous system. So when I was in medical school, I liked the brain mind problem, and I thought about psychiatry, neurology, neurosurgery. But I wanted doing a research elective before my senior year in a cancer lab, just for something different. And I studied mitosis and how cells divide. And everybody else in the lab in the early 70s got obsessed with the chromosomes. This is the dawn of genetic engineering and
Right,
all
right,
that.
right.
But for some reason, I got interested in how the microtubule spindles, the mitotic spindles that pull the chromosomes apart, knew where to go and what to do. They seem to
Oh,
have some kind of intelligence or maybe consciousness. I was interested in
boy.
consciousness also back then. I'd taken a philosophy of mind class and, did what we did back in the 60s. And so I was interested in the brain mind issues. And I just said, well, how do they know what to do? They seem to be running the show at that level. And at that time, the structure of microtubules was just being discovered by X-ray crystallography by Amos and Klug in the UK, and it was a cylindrical lattice of individual proteins, and and the geometry in the lattice was a Fibonacci geometry, which is a very mathematically evolved deal, and so I thought, and I was also learning about computers back then because it was the early 70s and they were new, and well, how do computers work? And I was reading about Boolean switching matrices and all this and that. And basically, you have a lattice of some sort and an interaction among the. And I said, well, maybe microtubules are computers, and the tubulins, the proteins, are the bits, and they interact with their neighbors. And so I finished medical school. I almost took a research career offer at Penn, but instead I wanted to move out west. So I came to Tucson and did a rotating internship, figuring, well, I'll figure out what to do. And I almost went into neurology, but I met the chair of anesthesiology who knew my interest and was desperate for residents. And he said, well, if you want to understand consciousness, figure out how anesthesia works, because we don't have a clue. And also he said he handed me a paper that showed that anesthesia depolymerizes microtubules, although it was quite a high dose. So depolymerization is probably, but it showed that anesthesia acts on microtubules. And then he said, it's a lot of fun and you'll make good money. And it's only a 2-year residency at that time. I said, okay, sign me up. So I did, and his name was Brunel Brown, my chairman and mentor for years until he passed in the mid '90s. And he kind of gave me free reign to. I had to publish and perish within anesthesia, but other than that, he gave me pretty free reign. So, but I wasn't doing. I did a few lab experiments. Mostly, I just did theory and modeling in the '70s and '80s and work with engineers and physicists, including at Los Alamos, and got to go to some of the chaos meetings and the nonlinear dynamics meetings and the cellular automaton meetings because somebody invited me to give a talk in the math department about my microtubule information modeling. And so I was showing how the individual tubulins interact and that you get these patterns moving. And somebody said, are you talking about a cellular automaton?
Yeah,
And I said, what is a cellular automaton? I'd never heard it before. And they explained it, and it turns out there's a meeting in Los Alamos coming up, and I sent an abstract in on microtubules as cellular, or normally meaning cell as a fundamental unit. So it was a way to express information in microtubules at a deeper, denser level than in neurons. So, for example, I read a book, Hans Moravec, Mind Children. Maybe you heard of it. He was at Carnegie Mellon, and he had this thing in there about transferring your mind to a computer, and it was based on 10 to the 11th neurons, 1,000 synapses, which gives you 10 to the 16th operations per second for the brain. And then Kurzweil picked up on this number later, and probably Minsky too. So 10 to the 16th, that was per second for the brain. That was their when we get that, give us a few more billion and we'll have a conscious computer and all you rich people can download your consciousness and live forever, which is what they seem to want.
Yeah.
And but if you calculate at the microtubule level, you have about 1 billion tubulins per neuron, and they're oscillating at various frequencies. That's a whole nother story. But 10 megahertz is a good number, and I'll explain why later. So 10 to the 9 times 10, it gave you 10 to the 16th operations per second per neuron. So, Kurzweil was saying 10 to the 16th for the brain. And I was saying 10 to the 16th per neuron times 10 to the 11th, 10 to the 27th. So I was going around to AI meetings and neural net meetings being a real pain in their butts because I was saying, no, your goalpost is way, 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 way down, 11 orders of magnitude further down. So, and they didn't like that, they said,
Oh, no,
go away,
no.
kid, you bother us,
Yeah,
you're
no,
gonna scare
that's
off
not good. Yeah, yeah,
sponsors. And that didn't really bother me too much. I kind of enjoyed being a thorn in their side. But one day, somebody said to me, "Okay, Mr. Wise Guy, let's say you're right. You have all this information processing. How does that explain consciousness? How does that explain feelings, love, joy,
yeah,
all these
yeah.
everything?" And this is basically what Chalmers coined as the hard problem 5 years later at the conference I organized. But so I was having the hard problem thrown in my face 5 years before it had been, but other people had said the same thing. Searle had a good argument. Nagel had a good argument. A lot
Right.
of people talked about this issue that computation did not automatically give you consciousness. And this person said, when he asked me, I was a little bit stunned. I was taken aback. I realized I don't. I'm a reductionist. And I don't have a clue about consciousness. But fortunately, he said, well, you should read this book by Roger Penrose called The Emperor's New Mind. And so I did. And it was really a, life-changing event because I didn't understand it all. It's so esoteric, but also the way Roger writes and uses his drawings and cartoons, you get the idea.
Yeah, right.
And the first half of the book was about Gödel's theorem and how consciousness could not be a computation. And he used Gödel's theorem, says basically a mathematical theorem cannot prove itself. You have to have something outside, like a mathematician. And Roger extended this to understanding, just conscious understanding of anything. And this is very much like Searle's Chinese room argument. The guy you get the
Right.
Chinese letters and you have a lookup table and you can translate, but you don't understand a bit of Chinese. And Roger's making the point that for understanding, you need something outside the system to judge for judgment. What that was, it had to be outside the classical system. And that's where he got into quantum physics. So the second half of the book was about quantum. And I was only vaguely familiar with quantum. But again, he used simple cartoons, including well, the first thing he did was, he kind of went over what is entailed by quantum mechanics, including quantum superposition. There's several mysteries, how things can be in multiple states at the same
Right.
time. Entanglement, how things can be connected over distance. Coherence, they can condense to unitary
Right.
things, entities. But superposition was, he's the only one who's actually ever tackled that, I think. And to do so, he went to general relativity.
Right.
And he said, okay, so big things like the sun curves gravity. So light behind the sun can be curved, and you can see it on Earth. And Eddington did the experiment in 1919, proved Einstein right. Eddington got his Nobel Prize, and they celebrated. And Roger applied it to Roger, being an expert in black holes and space time and everything else, applied it to tiny things. He said, well, a big mass has a big curvature, a tiny mass will have a tiny curvature, and is essentially equivalent to the curvature. So if you have a quantum particle that's a tiny curvature, then you can imagine that if it's both here and here, you have a curvature here and a curvature here in space-time. And so you have a separation in space-time geometry. And so Einstein's metric, the space-time continuum, actually separates. And you can imagine that if each possibility continued, each space-time curvature continued, you'd have the many worlds hypothesis.
Right.
And as you know, many people think,
Right.
well, what happens to the superpositions? And they either collapse, or they kind of get eroded by decoherence, or there's an objective threshold, or there is no collapse, and each possibility goes off and forms its own universe or evolves in its own universe. So Roger came up with another category, objective reduction, where the superpositions terminate. So instead of going on and forming their own universe, the separations are unstable and will collapse at time t to one or the other. T equals h bar over E sub g, where h bar is the Planck-Dirac constant, and E sub g is the gravitational self-energy, the amount of energy to pull a particle apart from itself or its space-time apart from itself. And when that reaches threshold, there would be a collapse to one or the other. And this was the real kicker. He said when that happens, there was a moment of now experience, essentially consciousness, a little blip or bing of consciousness at a tiny
Yeah.
microscopic level. So it was, and I think remains, the only scientific proposal for conscious experience ever put forth, other than emergence, hand waving emergence with some formula or another.
Right.
without saying whether it's firings or whatever it is that you're measuring or is being processed. So he took it to the space-time level, the most basic level. Don Hoffman may say, well, we have to go deeper than that. Maybe he's right, but I'll take the Planck scale for now for the basement level and worry about the amplitudahedron or whatever.
Yeah, where he's going. Yeah, yeah, right.
Yeah. Basically,
Okay.
Roger Penrose connected consciousness to the fundamental level of the universe, which created other problems like how do we connect over that? But I'll come back to that. And so not only was this collapse conscious, or in a micro environment where everything's random, it would be proto-conscious, because this would be happening in the environment, in the air, or even in a vacuum with space-time separations and having these little moments, but they'd be random and disconnected. So they wouldn't have meaning, they wouldn't have any memory, they wouldn't have any context, and they'd be happening everywhere. And but that sounds weird, but proto panpsychists say pretty
Right.
much the same thing, except there it's a property of matter, and then you have to explain how all those individual atoms combine, the combination problem in panpsychism. But this was different. It's what Chalmers later called pan-protopsychism or quantum pan-protopsychism. He didn't approve of it, but that's what he called it. And and it gave you consciousness. And the choices in the collapse were not random as they are if you make a measurement or in decoherence, nor are they algorithmic as it would be in a computer, but they had this other influence that had to be the outside influence, like in Gödel's theorem, which had to come from quantum. And that turned out to be an influence that wasn't random, but was what he called non-computable, which means it was influenced by what he called Platonic values em- uh,
Ooh.
embedded in space-time geometry. So that was
Could you
he didn't like
elaborate
that
on
one.
what you mean by Platonic values? And so there is a instantiation of Platonic values in the collapse of that you're talking about? I mean, is that what creates consciousness? Is a conscious expressing itself?
It's when the collapse occurs, it's a process in the fine scale structure in space-time geometry. And the way I interpreted it was that when the collapse happens, there's an influence. It's like a crooked
Oh,
roulette wheel. It's going to go to one rather than the other because of something intrinsic to the universe that's going to guide it and influence it. And you could make that sort of a religious thing, like the way of the Tao or divine guidance or something like that, or Plato's ideal forms or something like that guided our perceptions of what the world is and our actions, what to do by, if you're mindful and don't act reflexively, the way I think of it, you could be more mindful and
right.
and do the right thing if that's how you're wired. So and Roger didn't talk about the quality of the experience, but if you have that influence coming from space-time, I think it's the logical assumption is that since qualia experience has to come from somewhere, and I don't
Right.
think it's an emergent complex
Yeah,
problem
gotcha,
because
gotcha.
there's nothing complex about a toothache. So I don't think you need complexity for consciousness, and I think it could be very simple. But anyway, these OR objective reduction, objective threshold, quantum state reduction would be happening all the time, but they would, like I said, they'd be disconnected, so they'd be basically proto-conscious. Roger coined the term proto-conscious, and kind of like noise. And the way I look at it is if you go to the symphony and the musicians are tuning up before they play, it's kind of a cacophony. It's noise. Musicians tell me, well, they're actually tuning to the same something, but I
Right,
don't
right, right.
understand that. So to me, that's analogous to proto-conscious noise. When you hear all that sound that doesn't sound, it's discordant, and then they begin to play Brahms, Beethoven, the Beatles, whatever, and that's music. And I think that's what our brains do, and specifically what our microtubules do, is they orchestrate the quantum OR events that are happening anyway, and they put them together into something useful, cognitive, and experiential.
Could I just try to interpret this in a framework that not dissimilar to what Friston was talking about, but maybe this is a stretch, but I mean, so he was saying he'll say to make predictions is to the reason things alive is it makes predictions, and that it has a it self evidences itself in making those predictions. And so there's a very strong relationship between something being conscious and something being alive, and that so when you have those interactions, you're being conscious and you're being alive in the context of the observer of that kind of thing. I mean, and it may be an instance of that, but and so when you I mean, so when you look at sort of mitosis or something, you think it's that continuous process of preserving state and bringing and revealing itself and stuff. Is that analogous or is that off
Well, you said a lot there. If we go back to active inference and predictive coding, which I think is kind of very similar in the brain, I think it's really a very good thing to describe what's happening in the brain because, for example, we know that incoming sensory information comes from the back of the brain to the front. And from the front of the brain going backwards is the model of the world,
yes,
basically the
exactly,
prediction. So
yeah,
you
yeah,
got from front to
right,
back predict
right,
expectations and
right.
they come in where they don't meet, where they don't match. That becomes
Surprising,
conscious. And Earl Miller and his group at MIT have done some tremendous experiments in the last few years on basically predictive coding, where they find that the front to back waves, well, first of all, they're waves, and they're EEG waves, but there's something funny about them. We think they may be phase differences at a faster scale, but I'll come back to that point. But the incoming is mediated by gamma synchrony, and the feedback is slower, and the gamma synchrony is supposedly mediated by GABA receptors. And yet, if you look at if you put the subject, the monkey, basically under anesthesia, it's interesting. What goes away is the feedback from front to back, and the incoming stays active, including the oddballs. If you do a test where the animal expects A, and then B unexpectedly, the B would be the oddball, and the oddballs still come through. They're not affected by the anesthesia, and yet there's no consciousness. So consciousness is in the front to back. It's in the predictions itself, which override. Well, it can be over if you're awake. The oddballs will register, and you'll see something new, novel,
Right.
and you're quite cognizant of it. But if you're under anesthesia, the oddballs keep coming through, but you're not conscious of it because the feedback goes away. So it's in the feedback. That has to be but those are the same neural functions, the same receptors, the same neural activity that mediates everything else. So what's different about that? And I think that's the other problem, because I think both the predictions and the incoming are mediated not just at the neural level, but at deeper levels in the microtubules. If I can go back to the microtubules for a second, when Roger and I put this forth in the mid '90s, we got all kinds of crap from everybody. Everybody knows the brain's too warm, wet, and noisy. How can you possibly blah blah blah? And you guys are crazy. Go away. And because if you build a quantum computer in the lab, it's got to be at absolute zero temperature to avoid decoherence. And you're at 37.6 degrees, you're out of your minds. And, our first response was, well, biology's had a couple billion years to figure this out. So don't be so hasty. And, but then actually the answer, so anyway, I'll come back to that in a second. But, beginning in about 2010, this guy, who's now a good friend of mine, his name's Anirban Bandyopadhyay. He's an Indian guy working in Japan and at the National Institute of Material Sciences. He's one of these guys who, as a graduate student, got a position at NIMS, National Institute of Material Science, directly to have his own lab, skipping all the postdocs. He just gave him money, do what you want. You have I don't know how many years, and he did. And so he built an atomic computer, atom, something like that. But then he ran into our stuff and got interested in microtubules and did a bunch of studies on microtubules. He found that there are coherent oscillations in microtubules. So neurons work basically in hertz. EEG is 0 to 40, 100, maybe 1,000 hertz. And he found if you look inside neurons, you look at the level of microtubules, you'll see kilohertz, megahertz, gigahertz, and terahertz. And in fact, you see if you do the same experiment at different scales, you see resonant responses that are the same every 3 orders of magnitude. They repeat. And they repeat you can sort of see it in the EEG. You see it in kilohertz, megahertz, gigahertz, and terahertz. And the pattern is a triplet of triplets. So it's three peaks, and each peak has three peaks. And this goes, we don't know how fast and deep this goes. I mean, in biology, it goes to terahertz at least, maybe to petahertz. But there's some suggestion that this triplet of triplets is actually in the universe and is in oscillation. I mean, you see it in NMR at the atomic level, but Anirban says you see it even without even smaller than atoms. So there's a possibility it's kind of an underlying rhythm of the universe, but we're not prepared to say that yet. But we can say that microtubules have these self-repeating patterns, resonance patterns at multiple scales, which seems to mean that they are what are called time crystals. And this was something new to me up until a few months ago when Anirban started talking about it, and he came out with a paper saying that microtubules are time crystals. And so just from looking it up my own, there was a guy, Arthur Winfrey, in the 60s who suggested that there were time crystals in biology. And but he could never prove it. And then Frank Wilczek, who won a Nobel Prize for, I forget, strong force or something like that, he got the idea that time crystals could be in physics. He predicted it, and the time crystal, basically the ground state, the lowest energy state, is an oscillator. So in a way, it's a perpetual motion machine because you're starting with the ground state and you have an oscillator. But 0 point energy, and in biology, you're starting at KT. So there is energy there. But depending on how you describe it, it's a perpetual motion machine. And I got criticized from that for saying at a meeting by a pretty prominent guy that I fight with frequently. And he said, well, time crystals are perpetual motion machines. And I said, well, so's life. So that shut him up, at least temporarily.
That'll do it. Yeah.
So the time crystal idea, I think, is one that we're looking at taking very seriously because it would explain a lot. And we're actually, so I'm working now in astrobiology, where we're looking for the origin of life in organic molecules from an asteroid that my colleague brought back. So we're going to, Anirban's working on this. We're going to be doing these kind of studies on these same molecules. And there's a hint that there's time crystal behavior oscillators all the way down to single individual molecules that were found in a meteorite. So we haven't published that yet, but that's something we'll be actively looking at. So I think this may be a deeper problem than people realize.
Could you elaborate a little more on what you mean by a time crystal?
Okay, so a crystal like salt or quartz is atoms that repeat periodically in space. So you have a crystal, right. Spatial crystal. Time crystal, you have a dynamical system which has a resonant dynamical pattern, and that repeats over time, basically, not just periodically in time, but at different frequencies, different scales. So you have the same behavior in kilohertz, megahertz, gigahertz, and terahertz. Basically, the faster ones are buried in the slower ones. And I asked Anirban how he discovered this time crystal behavior, and he said, "Well, I was looking at the kilohertz, and then I noticed off to the right on the graph there's a tiny little bit of noise, so I blocked out the kilohertz and then blew up the next phase, which was megahertz, and I saw the exact same pattern. And when I did that, I saw off to the side a little what looked like noise, and I blew that up, and that was also the same triplet. So it's repeating at every about 3 orders of magnitude, every 3 orders.
Wow.
So that gives you a scale invariant hierarchy, kind of like it's a time fractal, basically.
Yeah, that's right.
And one of the problems in biology, and I don't know if in AI too, probably it should be, is how do you transcend scale? In other words, right. We're the size we are, elephants are bigger, and then we have little tiny things, and yet within us we have all these tiny things going on, and yet we're one entity and so forth. And the time crystal is the way to transcend scale, I think, because you have the same information or maybe consciousness repeating at all scales, from the terahertz and petahertz down at the angstrom level to hertz at our level, and everything in between, but periodically, so that it's the same information.
Whoa. But that's I think, looking at some of the work that Michael Levin's doing, he's sort of getting at the same thing, I do believe, and what he's talking about. And It's
Yeah, well,
not.
he's
Yeah,
studying electric fields,
that's right.
and he usually doesn't describe their frequency,
Oh, that's right. That's
uh,
right. That's right. Right. That's right. That's
and
right.
nor does he describe where they come from. I
No,
mean, people in neuroscience are talking more and more about ephaptic fields as opposed to, axonal firings and synaptic transmissions, basically electromagnetic fields
exactly.
mediating interaction. But the problem with that is electric fields tail off, I forget, by the distance rapidly. So you'd have to boost them periodically to get from one side of the brain to the other. And by the way, in gamma synchrony, 40 hertz, you have perfect zero phase lag synchrony across the brain, which is
Oh,
pretty much impossible to explain by neural transmission because of the delays and so forth. And if it's not neural transmission, and if it's not ephaptic, it has to be quantum entanglement to have zero phase lag synchrony across the brain.
wow.
So
Wow.
I think so I think neuroscientists need quantum without entanglement without realizing it. Another
That's right.
example is spatial temporal binding. So if you go back to sensory processing, vision V1 and then it moves forward, you get shape, color, motion, meaning of an object at different places and at different times. And yet you don't see something shaped, then it's color, then it's motion, then it's meaning. It's an airplane or it's a bird or it's a kite. You see, it's a kite. It's a yellow kite fluttering in the wind. You see it all at the same time, instantaneously. And entanglement occurs not only spatially, connecting things over space, but also over time, which is also very important for other reasons. So I think even mainstream neuroscience, although they won't admit it, need entanglement. This zero phase lag gamma synchrony thing, if you look, well, nobody understands it, so we ignore it. And that's the problem. They ignore these important things that should be looked at. So I think quantum is necessary for neuroscience for a lot of reasons, including consciousness and binding.
I think Chris Fields would make that point too, but I think that you're speaking the specific kind of mechanisms and microtubules as a way of explaining this, and the different the fractal continuity at different scales. I mean, if you have this time crystal phenomenon, that's sort of fundamental, right? I mean, if that's the case, right? I mean, it's not. It's a property of the universe,
Right.
as
Well,
it
then
were.
the question is, okay, so how does this happen in the warm, wet, noisy brain? So let's
Right.
go back to decoherence because the answer is hinted at by anesthesia, I think.
Right.
So,
The
as I
tryptophan.
mentioned, one of the reasons I went in anesthesia was to figure out consciousness. And the anesthetic gases, they were discovered in the 19th century initially as party favors because people,
Yeah,
they would sniff ether and have ether
right.
frolics or sniff
Yeah.
nitrous oxide,
Right.
laughing
Like
gas,
laughing
and
gas, the whole
if,
thing. Yeah.
if they breathe too much of it, they become unconscious. And somebody said, if they kept on breathing, they wake up fine, and they didn't vomit and aspirate that sort of thing. But generally, you'd go to sleep and you wake up. So somebody realized, well, hey, we could use this for surgery. In 1846 at Mass General, they did for the first time and with ether. So if you look at the anesthetics, so there's roughly a dozen gases that can be used as anesthetics, and they all have different potencies. And these two guys at the turn of the 20th century, Meyer and Overton, tried to figure out, where they were acting in the brain, what they were doing. And so each anesthetic has a potency compared to the other anesthetics, and that potency applies to all animals. So at equilibrium, takes the same concentration of a particular gas, let's say halothane, to put a flea, a worm, an insect, or a whale, or I don't know about a
No,
whale, but yeah, whale too, if you could get. But anyway, large animals, small animals, they all have the same potency at equilibrium. It may take a while to get to equilibrium, but in the gas phase, but once you're at equilibrium, it's the same amount to put you to sleep or a flea or an elephant.
it really is.
And that tells me the consciousness is the same in all animals and plants. Plants also respond to
Yeah.
anesthesia, and they have, they follow the sun and all that, and that stops with anesthesia. And that's also mediated by microtubules, by the way, in the where the root meets the stem, apparently. So
Wow.
yeah, plants have microtubules too. So each one has a potency. That potency holds for all animals. they wanted to know, well, where's it acting? So they tested their solubility. So in anesthesia or medicine or pharmacology, you give a drug, you want to know where it goes, and where it goes depends on its solubility. And there's 17 solubility compartments that pharmacologists worry about. And you can boil it down to two big ones, oil and water.
Yeah.
And as you know, oil and water don't mix. And then there's all these gradations, but just consider oil. So oil would be like the aromatic rings in proteins, for example,
Right.
or fats or membranes. But it turns out where anesthesia acts is in proteins. And so when a protein folds, you have these the aromatic amino acids with the rings like benzene or indole. And indole is in serotonin and all the psychedelics, and the hexagonal dopamine. So all the molecules with feelings and consciousness related. So they're nonpolar. Okay, they're oil-like, and they don't like water. So in a protein, the aromatic rings coalesce, and they get the water. They get rid of the water. So there's no water. So the thing about too warm, wet, and noisy. So forget about the wet, because in this oil-like region, there's water, and these molecules are optically active. So quantum optically active. They're fluorescent. So if you hit them with UV, they peter along, and then they give off another photon, lower energy. And when that petering along, that's actually jumping from different quantum states. And then they emit. So they're quantum optically active. And so they avoid the water. So what about the warm and noisy part? What I just described would work for like one little protein with aromatic rings. But if you then take that and make a polymer out of it, so it's a periodic lattice. This goes to something that Schrodinger first said in 1935 in his book "What Is Life," where he said that there needs to be a periodic or aperiodic lattice. Aperiodic means not perfectly regular. You want a little something off, and that could process information and be a quantum device. And he said that life needs that, and I think that was probably before DNA was discovered.
Yeah.
And then in the 60s and 70s, a guy named Herbert Froelich, whom I had the fortune of meeting, was a really interesting guy. He was a quantum superconducting expert, and he looked at biology, and he said that if you have a polymer, a lattice, like Schrodinger had said, where each of the components had a nonpolar region where you can have inducible dipoles. So he didn't want permanent dipoles oscillating, because that's charge, and that causes decoherence. But if you have an aromatic ring, so it goes to benzene. So let me go back to the basis of organic chemistry. Like when I was a pre-med, they said, well, if you want to go to medical school, you got to ace organic chemistry.
Yeah.
Ooh, difficult, you know.
Yeah.
So and it's all about the aromatic ring. You start with benzene, where you have six carbons. They have 3 extra electrons, so they form these electron clouds above and below the carbon ring. And that electron cloud is basically a space-filling volume of quantum superposition, because the electrons are everywhere. It's like a quantum object, and it doesn't do chemical bonds. It doesn't make chemical bonds, but the carbons can make chemical bonds, but the electron cloud itself only does quantum interaction. So if you take, for example, 2 benzenes, if you put it in bulk form, it's gasoline. It goes in your car, fuel. But if you put it in a planar sheet like graphene, you have interesting quantum properties. Or if you put it in a cylinder like a fullerene nanotube or a buckyball, you also get very interesting quantum properties. So if you have 2 of them, if let's just say two benzene rings, they will attract each other because the electrons in one will repel the electrons in the other. So you get a dipole over here and a dipole here. So you have 2 dipoles, and they couple and they oscillate back and forth. And so two benzene rings, but left by themselves, would just attract to the van der Waals radius and start rocking and rolling back and forth. That's probably the basis for life right there. You need at least 2 of them. So, but then if you have a nexus of those, as in one component of a polymer, in a periodic lattice, they're going to have long range interaction and couple, and you have quantum coherence over the whole thing. And that's what Froelich said. Froelich said that you're going to have these giant dipoles and the whole system can be a quantum system. In the microtubule, the lattices they go up, but they also go around in spirals, in three different spirals in the Fibonacci. So you have these interesting, I call them topological qubits, quantum bits that follow the pathway in the microtubule. And again, it's all in the aromatic rings, and the whole thing is oscillating kind of like a laser. In fact, it's a lot like a laser, where a laser is a quantum device at,
Right.
at warm temperature, because you have to pump it. So instead of the energy causing decoherence, it's actually pumping if you have a lattice that oscillates properly. So it takes non-coherent energy into it and converts it to a quantum coherent. So that takes care of the warm, because that's what's driving it, and it takes care of the noisy because you have coherent, and the wet's not a problem.
Okay, good to
So
know.
inside the aromatic rings, you solve everything. And in microtubules, you have an aromatic ring from one tubulin to the next, to the next, to the next. So you have mesoscopic or macroscopic quantum states in the among the aromatic rings along the whole length of the microtubule, which can be pretty long in your sciatic nerve. They're continuous, or at least mesoscopic. Actually, I think consciousness is probably in dendrites, not axons, but that's another story. Actually, it's an important story that I'll come back to. But I think that's how you avoid the decoherence problem, because in these aromatic rings where anesthesia goes, and anesthesia is very selective. The only thing it affects pretty much is consciousness. Thank you. Is consciousness. In other words, under anesthesia, the brain is still active, and we can actually measure evoked potential. We put a patient to sleep, like for spine surgery. So we want to make sure the surgeon is not doing something harming the spine, the blood flow, or something to the spine. So we have physiologists who come in and recording from the brain and the periphery, and they can stimulate the periphery and see the signals come through and say, okay, your spinal cord's okay, spinal cord's okay. Or they stimulate here and you get motor movement for the motor side. So we know the spinal cord's okay, but the patient's, so these evoked potentials are happening all the time, and yet the patient's completely unconscious. So that shows consciousness is anesthesia is selective, so it's only affecting the stuff in the quantum areas, because that's where the anesthetics bind. They bind by the same quantum forces, and they're attracted. They get out of the blood and water because they're too polar, and they go right to fat and membranes, and inside proteins. We have these aromatic rings, so they're attracted to the quantum areas, and that's the pharmacology of anesthesia. Well, it has a particular Hildebrand solubility coefficient of 15.1 or something, but that just tells you it's nonpolar aromatic rings, kind of like benzene, and that's where consciousness happens.
Well,
And in but you need a proper vesicle or vehicle, and I think that's the microtubules.
so so this is a major departure from the more traditional computational model, to say at least. And what does it
I've
take
heard
for
that.
some, well, what is it? So and you're getting more and more evidence, I mean, from a variety of fields. I mean, you're getting really empirical evidence to this, and to me, it seems to be make, and I'm not a total layman on this, but it just seems to make sense. But what is it? And this is where I'm sort of interested in the sort of the history of science. What does it take for something that radical in order to get acceptance, even when there's evidence that's coming out? I mean, do you have to create a warm temperature quantum computer or something on these principles? I mean, what?
Well, that's a very good question. And in fact, Anirban is doing exactly that. He is building a growing a warm temperature organic quantum computer in India. And
Whoa.
he's going to talk about that at our conference. We organize the Science of Consciousness conference, which will be in Barcelona in July. I'll send you
I'd love
information.
to go. I'd love to
Yeah,
go. I mean, this
great.
is just great stuff. I mean, this is, yeah, yeah, yeah. Yes.
But getting back to, how is this accepted? Not very well by others, because a lot of people are invested in their neuroscience. The brain is a computer, AI, and what's happened in the last few years, I think. Well, I can put it in terms like this. So we started this conference in 1994. There's been a kind of a divergence of opinions into two general areas. I mean, there's a lot of different areas, but two basic areas. One is the brain is a complex computer of simple neurons, where each neuron firing is a one or a zero. and the brain is a computer and no different from AI, and therefore AI should be conscious. And AI people love this, and I think they're pouring money into labs that do this kind of work.
I agree.
And those labs are sucking up to AI, and they don't want to hear from us because we're going to ruin the gravy train. So, and the other side are people who think that consciousness is fundamental and somehow connected to the universe in one way or another. And this includes, spiritual people like Deepak Chopra or a lot of people like that, Eastern philosophy people. But there's also a lot of physicists like Roger and who would doesn't like to talk about spiritual stuff at all, but also people like Nassim Haramein and many other people who think there's a fundamental connection and are trying to make the connection. So I'm in that camp. But so on the one hand, you have the brain is a complex computer of simple neurons, and the other the consciousness is fundamental, and I call my term is the quantum orchestra. I say the brain is not a computer, it's a quantum orchestra, because you have these frequencies, spatial temporal frequencies. So if you get the brain, you go into the neuron, which is roughly hertz, then you go to kilohertz, megahertz, gigahertz, terahertz,
Exactly.
down and down. And eventually, although this part's gonna be hard to prove, you can get to the Planck scale. The time crystals could go all the way down. Time crystal behavior, even without the biology, it could be in space-time. Nassim Haramein thinks that, and some other people think that. Honorbaum thinks that, but I'm just worried about now in the biological sense where we can go down at least to terahertz and see these oscillations. And actually, the terahertz is where anesthesia acts. Anesthesia blocks the terahertz oscillations in the proteins in the aromatic rings, and that slows down everything else. The classical stuff continues, but things that depend on the quantum stop.
But if it's an orchestra, then who's the conductor?
That's a good question. It's more of a jazz. It's more of a improv or a jam session. That's a very good question, but there doesn't need to be an orchestra. There doesn't need to be.
That's what I meant. The conductor. I mean, they conduct.
Yeah.
But
You don't need somebody doing that because it's more, a jam session or a improv or jazz, there's no conductor.
Oh, but they're synchronizing among themselves. I mean,
they're resonating among them. Exactly.
Yeah.
Yeah, and they're entangling and resonating among themselves.
So that would argue for a plurality. I mean, how does that tie into sort of John Archibald Wheeler's concept of physics as participation of multiple observing? Is that consonant with that perspective then?
Yeah, well, the interpretation of the measurement problem include the conscious observer, where
Right.
If you observe, consciousness causes collapse of the wave function.
Right.
And Dave Chalmers espouses that view for some reason. We're good friends, but we're feuding right now because he went over to the, he came up with a hard problem and talked for his whole, ever since I've known him for the last 20, 30 years, that, consciousness is a really difficult problem, So this all started at our first conference in 1994, where the first morning I had three philosophers. The first two were very famous. And at that time, philosophers read from their papers with no eye contact, no slides. And the interdisciplinary audiences go, they're all falling asleep, And I said, oh, crap, this is going to suck. And then Dave was an unknown at that time, a postdoc, and had lobbied me for a plenary talk. And I needed another. I said, okay, fine. So he gave the third talk. And fortunately, I'm glad anyway, he gave a fantastic talk. He got up there and said, we have all these problems like memory, attention, behavior, reportability, and they're difficult and people spend their lives on them, but they're relatively easy compared to the hard problem of why we have feelings, qualia, the term for experience and so forth.
Right.
And, he had this kind of rock star aura about him, his hair, long hair, and he pranced up and down. And he really rocked the place, I have to say. It was great. And at the coffee break, I'm going around listening like a playwright on Broadway, and I'm going, oh, the hard problem. So they really got it. That really galvanized the meeting, the whole movement, really. But then he kind of went over to especially in the last few years where AI has been coming on strong.
Right.
He said, well, I didn't really mean that. I think AI will be conscious. So despite all the LLMs, I still don't think it will be.
No,
So, but it's
yeah.
politically and economically wise to do that if you're in the field because the AI is pouring money into it.
If it's purely computational, but I mean, so there is the work. I mean, Chris Fields and Don Hoffman and others have done a sort of how to create conscious agents. There's a sort of formal basis of and can you have some kind of an agent that's interacting with a quantum field in order to actually assert its own sense of itself? I mean, would that be, I mean, is it, I mean, you're not reducing it to a computational model. You're actually, so you're just creating the conditions by which consciousness can emerge through those interactions or that communication between the classical and the quantum.
I
Is
think
that,
for agency or for causal action, you need collapse.
that
So
that's what I'm getting at? But yeah. So you
I
have
mean,
the
a field per se won't do it. I mean, these ideas of consciousness is a field. Okay, you
But then
need
that would be the
collapse for
interaction. I mean, that's where the agency emerges in the consciousness of the interaction between the two, the collapse of is there something exchanged between the classical and the quantum? I mean, there's information that's being created, patterns that are being created. I mean, I mean, one of the things I think like I mean, Chris will talk about a hologram running to it, but and there's also Susskind's notion of a sort of his whole concept of second law of quantum complexity. I mean, where you have it seems to be a principle that's driving this kind of interaction between the two. I'm just
yeah,
throwing
I
that.
don't like, some of my best friends espouse complexity, but
It's a circuit complexity. It's a concept of circuit. I mean, I know complexity gets overused. I mean, I mean,
yeah,
but
it's like an
it is.
answer when it's not really a solution to anything.
Well, what he's saying is that each state tries to understand every as a representation of every other state. In a sense. So that drives the whole process. So that'd be part of the interaction between the two. I'm just throwing these ideas out.
Well, I'll go back to Roger's idea because I think the collapse is key. And over the years, a lot of people have said, "Hey, you don't, because they don't like objective reduction, they don't like quantum gravity, and they don't like physics, and a lot of neuroscientists. So they'll say, the microtube, that's a great idea. Why don't you forget about that quantum stuff or forget about quantum gravity and just say it's quantum coherence, this and that." And I'm thinking, and you have a good thing going. You don't need Penrose. And I'm thinking, wait a second. He's got the only theory ever
No,
for what
I agree.
consciousness actually is. And he's a Nobel laureate and he's freaking brilliant in every aspect that I've ever heard him talk about it.
Oh yeah,
So
yeah.
I'm not gonna, leave him. And he tells me, yeah, people say, yeah, quantum gravity, you don't need microtubules. And he said, well, they're the best thing I know. So we stuck with each other, and I'm glad on both
Oh
sides.
yeah.
But I think you need collapse for consciousness. And you talked about so in the prior to collapse, it's all quantum superposition. Collapse, objective reduction, then you get classical states. That creates the classical world.
That's what I mean. That's
Yes,
what I was trying to say. That's what I was trying to
yes,
say. Yeah, okay,
yes.
yes.
At least
Yeah.
for a moment, then it happens again
Yes.
and again. But you're
Again,
creating
again, again,
classical
again.
reality, and that
Right.
happens with any collapse, including the ones in our brains.
So when you see, I mean, what kinds of changes in your field, or what kind of evidence do you think will move ahead this way of thinking? I mean, we go back. I mean, we look like was behaviorism was dominant. I mean, there were always and Chomsky wrote the critique of Skinner, and that sort of killed it. Is it? But how would this proceed? What kind of thing needs to be done to have this new notion of consciousness and
Yeah, well, we've come a long way, actually. I'm pretty
yeah,
pleased where we are, and however, well, a couple of things are happening. Number one, despite the giant suck up to AI on the part of neuroscience, I think there's less and less evidence that the classical neuroscience can explain consciousness. Uh, but that could be, well, that's your opinion. So, but I think the proof should come from anesthesia because we know anesthesia is selective for blocking consciousness, sparing everything else. So if we knew exactly what anesthesia does and where it does, what it does it to, whatever it is, that would be a big step forward. So when as much of a fan of microtubules as I am and was, and as an anesthesiologist studying anesthetic action going back to the 70s, back then it was all membrane. Well, first it was this that the lipids, okay, it's in the proteins. Okay, which proteins? So they spent In 1984, it was shown that anesthetics act on proteins. And everybody said, okay, which membrane protein, which receptor, which ion channel? Is it the sodium channel? Is it the GABA receptor? Is it this? Is it that? So they spent 25 years studying membrane proteins. And anesthetics bind to a lot of them. GABA receptors, they bind quite avidly. And also serotonin receptors, glycine, acetylcholine, others. After 25 years, the leading people in the anesthesia research wrote a paper led by a guy named Ted Eager, who was the undisputed king of anesthetic mechanism research. They wrote a paper in a prominent anesthesia journal saying, okay, there's no single protein or no single membrane protein or combination of membrane proteins that can account for anesthesia because of the Meyer-Overton correlation that indicated a unitary site of action, that they're all acting at the same one.
Wow.
And not all anesthetics bind to GABA receptors, for example. So that kind of ruled that out, nor anyone. And so you'd have to show a Meyer-Overton correlation for an effect on a particular protein. So anyway, in 2008, after 25 years of looking for membrane protein sites of action, Eager et al. Published this paper that said we need a new paradigm. This ain't working. I knew Eager. He tried to recruit me to go to UCSF actually, and I always wonder whether staying in. I love Tucson. I didn't want to go to anyway. So I knew him. Let's put it that way. And I wrote and I said, hey, microtubules, because there's evidence in anesthesia find some microtubules. And they totally ignored me. They just went on. They went to back to lipids and emergence and all this other stuff. But then we started getting evidence for anesthesia acting on microtubules. So, for example, there was just a recent study that came out. Well, first of all, a lab at Penn, Rod Eckenhoff's lab at Penn, took us after this. After they kind of said membrane proteins won't do it, they did a systematic review of binding, and they found, for example, that anesthesia binds 70 different proteins in a cell, in a neuron.
Wow.
About half in the membrane and half in the cytoplasm. And to figure out which ones are just, side effects or inconsequential and which are the mechanisms, they looked at gene expression. And supposedly the gene expression changes if it's a functional effect. And they found the gene expression in the tubulin, in the microtubules. So that pointed to microtubules. Although they didn't even pick up the banner. They didn't want to go there because they were getting ridiculed for even suggesting that. And then they did an optogenetics experiment where they had an anesthetic, a fluorescent anesthetic that it was an anthracene molecule that was anesthetic only when you hit it with UV light and it fluoresced. And they gave that to tadpoles who had transparent heads. And when they turn on they're swimming around, they turn on the UV and they go belly up. So they're under anesthesia. Yeah. And when they ground them up, they found that it was bound to microtubules. And they also did a clinical study where people on Taxol, mostly women, because Taxol is an anti-cancer drug for breast cancer, which stabilizes microtubules. So the microtubules can't depolymerize for mitosis. So it blocks mitosis. But it stabilizes the microtubules, and they found that people on Taxol needed more anesthesia. That the stabilization, or maybe just blocking the access of the anesthetic to the binding site. So that was another example of for anesthesia acting on microtubules. So fairly recently, a group, neuroscience group at Wellesley College, a small school, with a young professor of neuroscience with a couple students and a bunch of rats, anesthetized the rats. So they put them in a chamber and turn on 4%, I think it was isoflurane, and then clock how long it takes for the rats to fall over. So it's a very simple experiment, but it's the kind of thing that Meyer and Overton did, 125 years ago. And so they gave the rats a drug called apothelone D, which is a microtubule stabilizer, like the Taxol. And they found that when they did that, the animals needed significantly longer exposure to go to sleep. So the apothelone D, which was stabilizing the microtubules, antagonized the anesthetic. So, that's a very simple study, but it's pretty profound, actually. There's been absolutely nothing like that for any other protein, any other receptor. So even though it's kind of a simple study with a few students and a few rats, it's a fantastic study. And that guy I invited him to speak at the conference in Barcelona.
Fantastic. Yeah.
But in the meantime, my friends and colleagues, they're going the other way. They don't want, they've given up on Meyer Overton. They just want to talk about emergence and stuff like that, which has nothing to do with anything. So
Wow.
I think, I think, okay, what's going to change it? So I can't say too much about this because I promised honor bond, but I'll tell you what I think I can say. And that is that I said Honorbound, we got to see so Honorbound's discovered these you know kilohertz, megahertz, gigahertz, terahertz inside
Yeah.
neurons, and you can now measure from the scalp buried in the EEG, and he calls it the DDG, the dodecanogram, for 12 orders of magnitude. And the most robust signal from the scalp is megahertz, and you get a peak at about 10 megahertz. And he's published a couple of studies. He's finishing up and writing up a big study he did in India. He went to a hospital in India and got the whole hospital to help him in the operating room, measuring this megahertz as patients go under anesthesia, as patients are in the ICU, they're in coma, and as they die, and versus awake. And I promised him I wouldn't divulge the results, but I'm very happy about the results. Let's just put it that way.
Good. Okay.
And within a few months, this paper will be out, or we can talk about it. But let's just say that it's looking like anesthesia acts at this faster level and it slows everything down. So that's gonna change a lot, I think. There'll be some pushback but that's science. And so I'm pretty optimistic that within a couple of years we're going to be on top of things.
Oh, really? Well, I mean, the Templeton Foundation did a number of studies too on this, and then it was a certain confirmation as well. I think from what I know.
Templeton, yeah, they had this program, the Accelerating Research in Consciousness,
Right.
and they gave a lot of money, and they wanted adversarial collaborations.
Yeah.
And this
Yeah.
is something that Kavanaugh and Kahneman, the guy who won a Nobel Prize, came up
Yeah.
with these adversarial collaborations. And the idea is that you have proponents of one theory and then proponents of the other theory, and they meet and they come up with an experiment that will falsify one or the other.
Right. Right. No,
And
yeah, yeah.
I was worried about that right from the start. We had a meeting with the people from IIT, Integrated Information Theory.
Right.
So
Right.
Tony,
Carl
and
yeah.
we couldn't come to an agreement.
Yeah.
They wanted us. So we said, what we wanted to do is have microtubules and detect quantum effects. And we've said we'll be able to detect clear quantum effects, quantum optical effects in microtubules, and then we'll add anesthesia and see if it goes away with anesthesia. So that was our proposal. And they said, no, no, you got to do it in a live animal, in an attacked animal. And
Yeah.
what I had just described, doing this in a microtubule, would be done on a quantum optical bench, with zero vibrations,
Right.
all these things lined up. You've probably seen optical benches, lasers and stuff, and you sample. And so it's because you can't have vibrations from the table or from the
Yeah.
cars or anything.
Yeah.
So that's what we proposed. And then Christoph and Julio said, no, it's got to be done in a live animal. So let's do that experiment in a monkey and we'll put the monkey to sleep. And I'm going, well, we can't do it in a monkey because we've never even done it on a bench in a single microtubule. Well, what does that tell you? Well, it tells you more than what you got. So anyway, to make a long story short, we couldn't come up with a common experiment. I don't think their theory is even falsifiable. I don't think it's a real theory.
No,
I think,
yeah, no, I get,
I
yeah,
think it's
yeah,
bullshit. And I like those guys, but anyway, Templeton gave us money just to do our experiment.
yeah.
So the joint experiments with monkeys or with humans, they're costing 5 million each. And all
Whoa.
of those, and they did a couple of them. So IIT versus Global Neural Workspace, and it was all inconclusive.
Yeah,
All
Carl
this money
yeah,
just down the drain. They
yeah.
gave us 200,000 to do two experiments, one of which is complete, and the other one, I don't know, they're still farting around with it. But um, what we showed was, and we went to Princeton. We went to a neutral lab, actually Greg Scholl's lab. It was pretty skeptical. And but my colleague Jack Jasinski had a postdoc who wanted to go to Princeton, so he went to Princeton, worked in Scholl's lab. So it wasn't something we cooked up in our lab where we could, put the thumb on the scale. It was done in a skeptic's lab, and we were able to show room temperature quantum effects in a microtubule. And what we did was, what they did, but I helped design it. So you have a microtubule and you hit it with a UV, and then you label it, and so when you get a fluorescence effect, but not just in one protein, it actually propagates along the microtubule. So I think six tubulin lengths, which was farther than you can account for by classical. So this propagation had to be a quantum effect. So we proved that, and then we had two different anesthetics, a gas anesthetic and a soluble anesthetic, and both of them dampened the quantum effect in a microtubule. So we called our shot. We made our prediction, we did the experiment, and we proved it. And we published it in a good journal. But because it wasn't the big adversarial collaboration, it didn't get a lot
Right.
of publicity. And in
Right.
fact, what did get all the publicity was the failed experiment between IIT and GNW.
Yeah.
And Christoph and Dave, my two former buddies, they had made a bet 25 years ago whether they'd find the neural correlate of consciousness or not. And they made a big
Right.
stink about this at the
Yeah.
ASSC meeting and didn't even mention the fact that, yes, one experiment worked. They just talked about experiments that didn't work. So we got totally eclipsed. But that's because ASSC is totally towards the classical material
Right.
computational, and we've now gravitated more towards the quantum fundamental level. So and they're fueled by AI money, and we're just on our own. But I think with the anesthesia experiments, with honor bonds, EEG experiments, if you're measuring megahertz, it can't come from membranes. It has to come from microtubules. Like I said, between that and the more anesthesia experiments and some other stuff, I think I'm pretty confident actually that in a couple of years, we'll be in good shape.
Well,
Maybe
do you
sooner.
think, I mean, you can grow a quantum computer in room temperature? I mean, do you think that's an evidence that's something you can see in 5 years or so? I mean,
I
that
think,
have
yeah,
qubits, it
I
have,
think
sufficient qubits to do interesting computation?
we'll see. But I think Anirban says it's already working in principle. And, compared to building a cold quantum computer, it's
That's
pretty
what, yeah,
cheap. I mean, it's a
that's what
million.
I mean. That's what
Uh,
I mean. Yeah,
yeah.
yeah,
So if you can grow quantum computers, and
right.
you need to interface. So you need all these peripheral devices. So he's
Right.
already making a supply chain, anticipating demand in the future. He's pretty smart guy. So I think that's going to be really good. And other people are getting into that now too. But he was the
I,
first as usual.
When I heard Peter Diamandis, you know him, I mean, and the X Prize guy, and he was expressing a wish for, he said the next big thing is going to be room temperature quantum computers.
Well,
That's
that's
surprising. I
exactly what
mean,
Anirban's doing it at a place called IIT Mandi. India has these
yeah,
25 of these
yeah.
IIT, Indian Institute of Technology, and the newest one is in Mandi, Mandi, which is up in the Himalayas. And I first went there about 3 years ago. They got into some of our work and they invited me, and I've been there a couple times. And they said, where do you think we should go? I said, you should go talk to Anirban. And they knew of him, but he's kind of aloof and kind of hard to get a
Yeah.
hold of. Get a hold of, and they weren't really sure. I said, please just talk to him. So while I was there, we had a Zoom with him, and he was saying what he was doing. They got interested, and he eventually visited. And to make a long story short, they went to the Indian government and got money to build an organic quantum computer in Mandi, and that's what they're doing. And the Indian government is taking this very seriously because they see it as a way of leapfrogging the West. Forget about the cold con. We're going to go right to
Oh,
organic.
yeah. No, no. I agree. Are they the only ones doing that? I mean, that's surprising. Is that, I mean, there's not
No, I think
other,
other people's are doing, but I think other people are doing it now, but they were the first, I'm pretty sure.
no,
I'm
that's
not
really
sure exactly, but I think other people are catching on too.
fantastic. But that's funny. Well, I mean, just in sort of a wrap up on the sort of the implications. I mean, so this is a very different way of considering reality. It opens up a very sort of different kind of cultural forms. If you take the full implications of this, I mean, do you have any thoughts or comments on that? I mean, because
Well,
you start to extrapolate. I mean, it really upends a lot of conventional thinking. There's certainly
yeah.
reductionist materialist thinking, and the primacy of space time in a classical world is being the only explanation of things. Say no, no, no. Maybe this is it's only part of the yin and the yang, and there is this interaction at the edge. I mean, would you extrapolate on that?
Yeah. Well, it reflects on our place in the universe. and the nature of reality, and you know, who and what we are and why we're
Yes.
here. You know, we are a process in space-time geometry. So it's kind of like neutral monism, but where it goes to quantum and collapse goes to quantum classical. But it also brings up another issue. So I retired from anesthesiology 6 months ago. I'm now working in astrobiology, and
Okay.
we're looking for the origin of life. And the way I got into that is that because Rogers' objective reduction can happen in anything for proto-consciousness, including space-time, even without matter, but in certain kinds of molecules and organic molecules in particular. I was thinking about the origin of life and evolution because I've never been real happy with certain
Right.
aspects of evolution. Like for example, life started, and then I don't know how many millions or billions of years before there were genes. So if there's evolution, it's without genes. And how and why would these molecules start behaving in such a way to be purposeful and to survive? Why would they want to survive or care about surviving
Why?
if there weren't any feelings? It doesn't make any sense. It never made any sense to me. So I thought, well, could it be the feelings were there from the start? That and the only way to get to that was Penrose OR, because most people say consciousness is complexity. So, you know, the
No.
brain started doing, he started doing all this
Right.
complex information processing.
Right. Right.
You
Right.
got to you
Go
get
brain. Yeah.
consciousness.
Right. Right.
Well, and some people say, well, that's fairly recently with language and tools. But that didn't make any sense to me. If you look at an organ, a worm, it's doing stuff because why do we do anything? Because it feels good, right? Even us, okay, we delay our
Right.
gratification, we go to school, we get a job, blah, blah, blah. But still, it's all what makes us happy, right? And so I thought, well, that would have been a good instigator for the origin of life. So I wrote this paper. I was asked to write a chapter for a book on human nature, and so I thought I'd do
Hmm.
something different. And I wrote it. I wrote this paper, "The Quantum Origin of Life: How the Brain Evolved to Feel Good." I put it out there. It's a chapter, so it's just I figure, well, it's out there. Nobody seemed to notice at the time. About 2 years ago, when I'm thinking, okay, what am I going to do when I'm done with anesthesia? Because I want to keep doing. So I was just going to keep doing research in consciousness and so
Okay.
forth, so on. So I heard from a guy, a planetary scientist at my University of Arizona, Dante Loretta, who was in charge of a 20-year NASA mission to an asteroid to bring back organic molecules. Well, the flight was 7 years. It was 20 years in the planning. And he was actually young when it started. He was the assistant to the head guy who got sick and passed away, unfortunately. And at a fairly young age, Dante became the acting chief and ran this NASA project. So when he contacted me about 2 years ago, they had landed on the asteroid. Well, they don't actually land. They kind of scoop it up, because if they land, they go right through it. It's just a bunch of rocks, and they come out the other side maybe. But they had successfully, and showed me the videos. Amazing what they did, actually. It's
Wow.
actually a simulation of the video because they didn't have a camera. Anyway, they had this or they knew they picked this asteroid because it was very dark, which tells them it's carbon, and it was also possibly on a collision course with the Earth, which gave them another reason, for defense, for
You
NASA.
get
So they picked
funding.
this
Good funding.
asteroid.
Yeah.
It
Yeah.
took 3 years to get there. They actually circled it for a year to survey where to land and measure it and all this and that. And then one fateful day, they basically had one shot. They went down, they scooped it up, they got the lid closed, and they headed back to Earth. So they knew they had something. And this had only been done twice before by Japanese on a different asteroid, Ryagu, and they got 2 grams of organic material. And Dante's project said, we hope to get 60 grams. And they got
Wow.
120 grams. So
Wow.
it was a big success.
Big one.
But they still don't know exactly what they had. And then last September, the thing came down in Utah, and it was on NASA TV. Dante went up there. I could see him, going to the thing, and he has a great picture of him looking at this thing. It just came back from this asteroid. Well, before that, so he contacted me about 2 years ago. He said, he said who he was, and he said he read my paper, The Quantum Origin of Life, and wanted to talk to me about it. He said,
Excellent.
he had promised NASA that they would find something related to the origin of life. So while the thing's coming back, he had to figure out, well, what are we gonna do now? So he said, he had independently thought, well, maybe consciousness was there first. And he went to the literature, and the only paper he could find on this was mine. He
Whoa.
emailed me, and he said, I'd like to meet with you. And not only was it mine, I was at the same university. So across the street at the medical school. So I said, well, yeah, I'd be happy to talk to you. So I went over and he told me the whole story. I was kind of blown away by it. He showed me this video and he said, do you want to work on this? I said, hell yeah, I do. In fact, I'm retiring from anesthesia. I'd be happy to work on this afterwards. So, and I figured, I have a pension. I would just work on it. But we actually got funded and from a private source that had been funding my work. And he's been giving us money. So I'm actually getting paid for it. And I brought an honor bond because he can do studies on these molecules that nobody else can do. And for example, now that we have the samples, you can see that one of the key finding in the samples are what's called nanoglobules. So in the origin of life story in the primordial soup, supposedly we had they had these micelles, operant micelles that were kind of like liposomes, with a shell and then something inside, which included organic molecules, aromatic rings, which could have the fluorescence and the quantum. So in my paper, I said, well, maybe they got together to start having Penrose OR and having conscious or proto-conscious moments, some of which would be pleasurable, and they would arrange themselves to optimize the pleasure. And so that's what I proposed in this, and that life started to optimize pleasure or to avoid displeasure. And that was the basic idea. And I explained how it could happen with OR. I talked to Roger about it, and Roger said, "Well, I don't know if you can get that many." And so he was skeptical, but typical Roger said, "Well, go for it." So I did, and I wrote the paper, and then Dante picked up on it, and he had a similar idea, but didn't have a mechanism because he wasn't familiar with Penrose OR. And so we wrote a position. So the guy gave us funding for a year. He said, write a position paper on what you're going to talk about. And we did, and we published it in actually a kind of a popular piece for IAI TV. I'll be happy to send it to you. But in the meantime, anyway, that was good enough for the guy to give us $1 million to keep going. And so that's what we're doing now. Right now, so what I'm working on now, which I'll get back to later today and over the weekend, is a paper for meanwhile, I'll get back to it, but Dante's been writing papers like crazy from all this material they got. So this is only one small facet of the whole thing. He said, I don't know, 6 Nature papers on this, something like that. But the paper I'm writing, well, we're all writing it, but I'm the lead on it, is the consciousness first proposal for the origin of life. And so we're going to take the pop piece that we wrote and beef it up scientifically. And hopefully we'll have some results. We already have some results showing like oscillators at the ground state in molecules. So even before we got the Bennu samples, we had there was a meteorite that came down in '69 in Australia. And there's one molecule that they found, which is this branching tree-like, looked like a bouquet of flowers, except instead of flowers, there were aromatic rings on it. And it was a 3D bouquet, you might say, but then pressed under glass, so it was flat. So you just see a 2D. It's been in the literature for 50 years. And we looked at it and we said, I wonder what that would look like in 3D, And so we sent the molecule to Honorbon. He did a 3D computer simulation of it. And, the aromatic rings, just like protein, folded, and we got a completely different 3-dimensional image that kind of looked like a cigar or a dimer. And then I shouldn't say too much, make too many claims here because we have to. But anyway, the dimer, it seemed to be wanting to form a microtubule. The dimer would align side by side and then curve into a cylinder. And we said, this is crazy. This goes against, it's no ribosome. How can you have a protein? But it's not really a protein. It's kind of like the schematic, the intelligence system of a protein, just the aromatic rings. And then we found the time crystal behavior at the most basic level. So we did a poster on that, but we haven't published that. But we're going to continue with that. We're going to do the same thing with the molecules from Bennu. And tubulin, in evolution is kind of a strange story because
Yeah.
The origin of life, the controversy about intelligent design, which usually is meant to imply God comes in and puts his finger on you get life.
Yeah.
But it could be a bottom-up intelligence or consciousness first, actually.
Yeah.
That's kind of what I think. 2 billion has never fit in the evolution because uh, intelligent design people have used it as example. Why would a protein evolve to make a giant structure that you don't know about in advance, or it doesn't know about it? It's kind of like, why would a brick evolve to build a skyscraper?
Right.
And so there's always, and now we're seeing that, well, maybe this came from a lot earlier than before. So we have a lot of possibilities. Maybe it's just a pleasure-seeking molecule or something like that, or maybe it's the fullerenes. So organic hydrocarbons. I was telling you about, so the aromatic rings in aromatic amino acids and everything else. Organic molecules, stars, young stars put out aromatic rings. I mean, all the aromatic rings came from asteroids, which came from stars. And by the way, it looks like Bennu that we're studying, the asteroid that Dante picked, actually was formed by a larger thing hitting a planet that or something that had primordial soup on it, because we have everything in the Bennu to make life. Everything. I've got a list somewhere, but everything you need to make life is in this one asteroid, including these nanoglobules, which are fluorescent inside. So we're trying to Anirban's trying to probe them without breaking them. And he's doing non-invasive studies using cloaking and tunneling and all this and that. So hopefully we'll have some answers. And then if we get, so we've defined, so you could say, okay, you guys are looking for the origin of life. How would you know if you found it? What are you looking for at the molecular level that would tell you it's alive?
Right.
Because most explanations are functional, like adaptation, homeostasis, this and that. Well, you're not going to see that at that level. So we've identified what we call primitive signs of life, or signs of life, including like the time crystal ground state oscillator, coherent oscillation between aromatic rings. And we've got actually 10 of them. I forget the others. So that's what we're looking for on these molecules. We wanted to put out what we're looking for first. So if we find them, we could say, hey, we predicted this, rather than after the fact saying, yeah, this is what we want. So we're going to publish that in this paper, and well, it's already out in the poster, but we're going to publish and the pop piece. But this is in for the Astrobiology journal. So I guess it's a whole special issue on theories of origin of life. So we'll be up against all the others. So that's what I'm working on.
Well, that is a worthy task.
Pardon me?
That's a worthy task. That is a worthy undertaking.
Thank you.
And to know that you've answered it would be that would be unequivocally prove that.
Thank you.
Yeah, no, that's fabulous. Well, I really appreciate you taking the time here, and this is a really informative journey and a lot to digest, as to say.
Thank you, and thanks for absorbing it. I think you picked up on it, and I appreciate that. And
well, it's phenomenal. I just think it's very transformative, and it's just interesting to me from a history of science. What does it take for these ideas to overcome resistance within other groups? I think in the note I say Max Planck says science progresses one funeral.
Exactly.
So what is it? The social construction that doesn't allow the recognition of it. How do you move beyond that? Anyway, so this is great stuff, and thank you very much.
Well, thank you. It was a pleasure talking to you.
Okay. All right. Take care. Thank you.
Bye-bye.